Well, hi, everyone. Welcome to our symposium on the role of the human thalamus with insights from functional neuroimaging experiments. The other three presenters and I are really excited to share some of our recent work on this topic, and we hope that you enjoy some of the insights that can be gleaned by thinking about the way that the whole brain intersects, the way that the cortex interacts with the thalamus. But I'm actually going to start at a completely different place, which is to talk about porphyrin rings. And so what are porphyrin rings? They're this really complex macromolecule composed of carbons, hydrogens, and nitrogens. Specifically, they have 4 modified pyrrole subunits interconnected at their alpha carbon atoms by a methine bridge. Yeah, I had to look that up in Wikipedia. So you're probably wondering, why on earth is Mac talking about porphyrin rings in a symposium about the thalamus? The reason I bring them up is that porphyrin rings are actually really common in biology, and they have a really special function, which is that the way that they're organized, the free electrons on all the nitrogens allow the molecule to bind a metal cation, here in this case zinc, but in other cases iron, such as in the case of hemoglobin. And by binding that cation and having this special property, it actually imbues that macromolecule with particular functions, like the ability to bind oxygen. And I think there's actually a really nice analogy to the thalamus. So this is an area we often think of the functioning of the brain, particularly the human brain, as really synonymous with the structure of the cerebral cortex. The outside structure of the brain, it's obviously expanded. You can just see it by eye over evolution, but it's intricately and hopelessly interconnected with the function of the thalamus. And the thalamus actually imbues the cerebral cortex with a number of its really important properties. And so hopefully today, my co-presenters and I can convince you of the importance of this structure for thinking about the function of the entire brain, and particularly the function of the cerebral cortex. It's been known for some time that we can use neuroimaging to map elements of the thalamus. On the left here is a picture from a really beautiful paper in the early 2000s by the Oxford group, using diffusion connectivity to track pixels and voxels within the thalamus and their connections, white matter connection strength to different regions of the brain. And you can really clearly see this property where the entire cortical mantle is interconnected with the thalamus. Traditionally, the connection was thought to be of a relay kind, or often described as a driver type, which you can see on the left-hand side in blue. But there's actually an admixture of different principles of connectivity that exist between the thalamus and the cortex. On the right of the right-hand diagram, you can see this red structure, which is projecting in a much different fashion, much more diffusely, and to different parts of the cortical circuit, which is called a matrix type. And we think that it actually imbues the cortex with completely different computational benefits and properties. So in my talk, which will come first, I'm going to speak about this diversity of thalamocortical projection types. On the right-hand side, the core type in green, and on the left-hand side, the matrix type in blue. And we can try to think about what kinds of benefits and costs come from organizing the system like this, and what kind of computational signatures might we expect to see if we start to interrogate the way that the thalamus can interact with the cortex. In the second talk, my colleague Doug Garrett from the Max Planck is going to speak about temporal variability and network integration and dimensionality, thinking about the role that the thalamus, which has a much smaller number of cells than the cerebral cortex, might play in shaping, constraining the kinds of functional modes that can occur in the cerebral cortex. You can imagine that there's going to be a strong relationship there, but working out exactly how those systems interact is a really important open question in neuroscience that Doug will speak about in his work. In the next talk, Hwang from Iowa will speak about the relationship between network topology and the thalamus. Now here on the left, I'm showing a diagram from a really beautiful line of work by Louis Puelis that doesn't relate directly to Kai's talk, but I think there's a really interesting corollary. If you look on the left, the map is of a developing embryo in the brain if you start from the right-hand side in green, the MES stands for mesencephalon, which is essentially the midbrain in a developed mammal. If you move forward up the chain, you see in pink the diencephalon, which is the thalamus. Further forward and down is the hypothalamus. And then up to the left, that big bulging area called the pallium is essentially where the cortex, the striatum, the pallidum, the hippocampus, the amygdala, insula, all the regions that we typically talk about live in that bulge on the left-hand side. And the important point to note is that topologically, the thalamus is sitting at a really crucial point where it's integrating information from the periphery through the spinal cord and the hypothalamus with the input from the telencephalon and the cortex and striatum, amygdala, and hippocampus. So it has this really topologically central role through development and in the organization of the structure of the brain. Now, Kai's work isn't really working on development, but what he's trying to do is understand the way that the thalamus interacts with the rest of the brain in terms of thinking of the brain as a network. And he describes the fact that the thalamus often has a much higher diversity of connections, which we can quantify with the participation coefficient, and also that the matrix thalamic nuclei, which here are defined as the higher order thalamic nuclei, have a slightly higher participation coefficient or diversity than the lower order or core type nuclei. And Kai will talk more about the implications for that organizational structure for things like cognitive function. And then in our final talk, last but by certainly means not least, Laura Lewis will talk to us about some really exciting work she's doing, pushing the limits of fMRI and trying to combine different vantage points, EEG and fMRI simultaneously to understand the interaction between the thalamus and the cerebral cortex, and really in light of its relationship with things like arousal and the difference between sleep and wake states. So I'm really excited to hear what Laura's going to talk about today and how it will intersect with the rest of what we're working on. So with that, I'd like to say, please enjoy the talks. We're going to be around virtually at the end to answer questions, I hope. And if not, you can find each of us online to ask questions if you have anything to ask after the end of the talks. Thank you very much. All right, so I'll be speaking today about the role of the thalamus in shaping and constraining whole brain functional connectivity. So as I said in the introduction, many of our explanations of the organization of the brain, particularly the human brain, are shaped around the way that the cerebral cortex is organized. This is a perfectly reasonable way to look at the brain. You can go back to the pioneering work of Marcel Messelum to look at this idea that there's a spatiotemporal gradient of different connectivity patterns and interactions that underpin the functionality of the brain. People in our community are well aware of the idea that you can take resting state data, apply some dimensionality reduction to the data, and recover these low dimensional spatiotemporal gradients that betray this functional signature. Here on the right, I'm showing a map from Dan Margulis and Johnny Smallwood's really beautiful work from 2016, where they did such an analysis and showed this low dimensional gradient that really maps back quite elegantly to Messelum's work from the late 90s. Because of this structure that is really sitting right in front of us, and also a number of other corollaries from cortical lesions that we might see in study of neurology, many of our models of how the system functions are really focused in on the cortex. This is a map of the traditional Rao and Ballard model of predictive processing in the brain, where there'll be a prediction from a granular region of cortex that comes back and is met with evidence that comes forward from the sensory input. And then the mismatch between those two signals is propagated forward as a prediction error. As I said, most of these models are really depicted at the cortical level, but don't necessarily embrace the kinds of architectures that are imposed by subcortical influences from areas like the thalamus. So if you take anything away from today, I'd like you to just not forget the thalamus. Let's keep this in mind when we're trying to think of the functioning of the system. So in my introduction, I talked about this really classical example of thalamocortical interactions. The lateral geniculate nucleus of the visual system is the one that many people think of, but the idea is this core type of processing where there'll be an input from, let's say, the sensory periphery, maybe the cerebellum, it'll then project in a targeted way to the granular layers of the cortex, which are intratelencephalic neuron, which means they project only within the cortex, and they project usually forward towards more agranular regions of the cortex. And this is thought to be something like a relay, although there's some recent work that sort of takes that into account and maybe suggests that there's some more computation going on in this layer. But anyway, this is the typical way that we were thinking about the thalamus. And this is a standard picture from any psychology or medical textbook, where you have input via the retina that comes via the lateral geniculate nucleus, and then sort of passes on in a relay fashion the information to V1, the visual cortex, where it then goes and categorizes the input that it's seen, and it decides what to do with it. But as I said in my introduction, the core isn't everything. So if we go and actually look at the connectivity of the thalamus, we actually see that there's a separate connection type, which is intermixed in between all of the different core projections. And there's actually every single nucleus that we would define in the thalamus actually has a blend and mixture of these different types of cells. And this matrix-like cell actually projects in a much more diffuse pattern and hits the supragranular layers of the cortex. So these are the big, thick, white matter-rich areas of the cortex that have a lot of projections that interconnect cortical regions. They'll either have projections coming forward and hitting onto areas in a higher region, or projecting back and hitting onto the apical dendrites of pyramidal cells in those higher regions. In those lower regions, I apologize. And we call those the matrix cells. This is a terminology that Ted Jones adopted back in the early 2000s or late 90s. And rather than thinking of these as the only things in the thalamus, it's better to think of them as two ends of a spectrum. If you look in between, there's many different weird and wonderful combinations of these different patterns. But the idea is, on one extreme, we have really targeted, specific input to the granular layers, and on the other end, we have really diffuse input across cortical regions to the supragranular layers. Now, rather conveniently, these regions also, these different cellular populations in the thalamus also have their own genetic expression of different calcium binding proteins. So on the right here for the core, we have the calcium binding protein parvalbumin, and on the left for the matrix, the calbindin. They also have calretin as well, which is calb2, but calbindin was the sort of main calcium binding protein that's used to distinguish between the matrix and the core cells. So we're really interested in how these different populations in the thalamus might interact and intersect with the kinds of patterns that we see in the cerebral cortex, particularly during the resting state and in tasks. And so what we did is we turned to the Allen Brain Atlas. And so what we were able to do there is find the mRNA expression of these different calcium binding proteins within the thalamus. And we looked for the relative loading of either calbindin or parvalbumin, and then basically put that weighting onto a voxel within the thalamus from a data set, a 7T data set with 60 subjects that was collected by some collaborators at the University of Queensland. What we could then do is extract those time series and relate them back to the kinds of patterns that we saw in the cerebral cortex during the resting state. And so this here is now a map of the correlations that we saw that were either stronger for voxels that loaded onto parvalbumin, they're shown in green, or voxels that loaded positively onto calbindin, they're shown in blue. And you can see from the plot on the bottom that the regions that were strongly loaded onto the parvalbumin population were typically those that comprise the visual system, the auditory system, and part of the somatosensory system in the parietal lobe, whereas calbindin was much more strongly coordinated by areas in the default mode network, frontoparietal network, and the salience network. And I think if you squint, you can actually say that this actually looks quite familiar to the primary gradients that were discovered, inspired by Mesalem, but were really sort of unpacked and discovered by Margolis and Smallwood, which means that we have a Margolis alert. We've found something that looks like a low-dimensional cortical gradient, but it's due to or related to the influence that of a genetic population, a genetic gradient that existed within the thalamus. So we can directly relate those to one another. Here I've just plotted the primary gradient from the Margolis study, which I was able to conveniently download from the NeuroVault Atlas, which was really great in unthresholded form. and I extract regional signatures of each of those different loadings from the Schaefer atlas, and I can correlate them together. And I see a really strong correlation here, correlation of 0.77, which is very, very, very significant. One concern we had early on is that because these patterns are so low dimensional, that it might be that just by being in the same parts of space within the connectome, we might find an inflation of our false positive rate. So we conducted what's called a spin test, and what you do there is you take your data, you put it back onto the free surface sphere, you spin it around 5,000 different times, and then re-extract the data and calculate a new correlation. And that gives you essentially a null distribution of how likely you would be to find the correlation just because of how smooth your data is. And essentially, the results survived that correction, which gave us some confidence that even if we take into account the spatial smoothness of our data, we have a correspondence between genetic expression in the thalamus and this low-dimensional correlation structure that is present in the resting brain in the cortex. We were also really interested in whether or not these patterns tapped into another really interesting phenomenon in the literature, which is that there's a hierarchy of timescales within the brain. So this is really beautiful work by John Murray and Xiaojing Hwang with Henry Kennedy, where they were actually able to show that there's a hierarchy of timescales across the brain that maps from sensory to more associative cortices. And so what we could do is we could calculate the Hurst exponent, which is a measure of how long the timescale is in a particular region. And we found a positive correlation between the extent to which the system was coordinated by the Calbindon-rich matrix system and the timescale that we were able to estimate indirectly from the cortical time series, suggesting that the matrix cells were actually able to imbue the system with a longer temporal scale. We also thought that based on the pattern of projections to the supragranular regions of the cortex, that we could think of the impact that the matrix cells were having as flattening this conceptual attractor landscape. You can imagine on the left-hand side, when the ball's blue, it's trapped inside a well, it's stuck into whatever process was ongoing. But if we were able to flatten the landscape, as you can see in the middle panels, now all of a sudden the ball can drop into another well and perform a different function. And it might be that having this diffuse projections gives you the ability to actually mediate these different flexible state-space dynamics. And so we quantified that by looking at the variance of the dynamic connectivity matrix over time. And we found, again, a positive correspondence between the regions of the brain that have a high variance, a high flexibility over time, and the regions that are more likely to have this Calbindon matrix-like property. In other previous work that I don't have time to really dig into today, we found a correspondence between the medial thalamic nuclei, which are really matrix-rich, and the engagement of a low-dimensional manifold here, the description of the brain in low-dimensional coordinates during a complex cognitive task. And so we think that there really is a strong correspondence here between the kinds of architecture in the thalamus and the way that it can actually shape and constrain the patterns of activity, low-dimensional activity that emerge in the cerebral cortex. To develop a more mechanistic understanding of what's going on, my postdoc Eli Muller has been conducting some really exciting work using a neural mass model. So the idea here is that we take something like a traditional corticothalamic neural mass. Here on the left, you can see pyramidal neurons and interneurons in the cortex that are then connected within a loop to the relay nuclei within the thalamus and the reticular nucleus inhibiting them. And so if you come back to my original starting point in this talk, this is really reflective of the kind of classical way of thinking about the thalamus as this recurrent loop of connectivity with the relay input. And what we wanted to know was, what if we imbue some of this diffuse connectivity into the system? What if we take a dial and we turn up the amount of diffuse connectivity, which could be thought of as equivalent to this matrix population, and we ask what changes in the network dynamics? For simplicity, we used a toroidal structure, which means that we don't have any boundary conditions. But obviously that is not exactly reflective of the way the brain would look, which will have much more like a rich club structure and might have a degree distribution that's scale free. So this is really sort of a simplistic model, but it does have a lot of the characteristics that we're interested in changing here, this diffuse coupling parameter, without a lot of the other sources of noise that might come from having a more complex structural connectome. And Eli's hypothesis was that as we changed the amount of diffuse coupling, it would be equivalent to changing the temperature slowly of a cup of water. And what happens in that case is just because of the distribution of the energies within all of those different water molecules, at a particular point in time, as the water temperature is increased, as you say, heating it from below with a Bunsen burner, a small population of those water molecules are going to think that it's even hotter than it actually is because they've been bumped into by their surroundings, and they're going to be much more likely to jump up. But the important point is that because the rest of the water is actually not that hot, it keeps the whole system from transitioning into a new state, but instead allows there to be a separation of states. You can have both a cold and a hot state at the same time. And so the idea was, can we change this diffuse coupling parameter and see if we observe these kinds of phenomena within a neural mass model? Essentially, yes, we could. So as we turned up the diffuse coupling parameter, we saw that the system transitioned from this blue subcritical zone to a saturated zone where all of the network had jumped up to a higher attractor. This would be like the little water molecules all turning into gas. But then right in that middle zone, what we call the quasi-critical zone, there's this ability to have this mixture of states where you can have both high activity, but then a large amount of the system is still stuck in that low activity state. So you can actually have the flexibility and the integration and segregation in the entire system. In terms of the neural mass model, the big thing that was changing was that on the left side in the subcritical zone, the reticular nucleus was blanketing the sensory relay nuclei and the thalamus, but on the right-hand side, the sensory relay nucleus had sort of won out and had more influence over the cortex. We found lots of interesting phenomena that I don't have a huge amount of time to go into. Essentially, in that quasi-critical zone, you could see a real separation of timescales. It really was as if the attractor landscape was flattening such that you could get to a new zone. But if you went too far, the system would become saturated and look something more like an epileptic model. You can also then go back and calculate all kinds of different measures that we're interested in brain imaging data, like the participation coefficient, which we saw increased in that quasi-critical zone. We can look at time series variability, which increased. Regional diversity is a measure of the diversity of the connectome at each point in time. And you can see there's a really interesting nonlinear relationship between those two. And on the right-hand side, the percent explained variance shows that the brain becomes relatively low dimensional in that quasi-critical zone. We can then go back and take each of those measures and calculate them on task data. So here, this is from the NBAC task in the Human Connectome project, comparing it to REST. And you can see that each of these signatures is actually reflective. Each dot is one subject. Each of these signatures is reflective of a change in the state space dynamics. And then if you map that back onto the brain, you can actually see that we can think of task states as in a more quasi-critical zone than rest states. On the left-hand side, each of these points to histogram using a model fitting algorithm to determine where was the best fit in terms of our model for this data. On the right-hand side, I'm fitting each of the different values back onto the cortex. And there's actually a positive correlation between that measure, weak positive correlation admittedly, but significant between that measure and the amount of, and the CPC map that we identified from the resting state. So in summary, we think that it's really important to think about the thalamus and its impact on the cortex when we're thinking about these whole brain operation processing modes. And that in doing so might give us some insights into what kinds of computational benefits come from organizing the system in this way. And with that, I'd like to say a huge thanks. I've bolded Eli and Brandon Munn, who's one of my other postdocs on the right-hand side, who did the lion's share of the work here. And with that, I'd like to say thank you very much.
Thanks very much to the organizers and to the OHBM for giving us a chance to present the symposium on the thalamus. And I'd like to talk to you today about the work we've done in that regard. Just a brief snapshot of the work we've pursued over the last 10 years. What we tend to find is that younger, higher performers typically exhibit more moment-to-moment variability in their brain signals within and across brain regions and tasks. And we see this as a broad-scale reduction of dynamic range with increasing age and poor cognitive performance overall. We also see that signal variability is parametrically modifiable and that it often rises with cognitive load, especially in high performers. So it is movable within person. And you could say that this past stuff has focused on what we might call local variability because we're measuring it in fMRI, for example, in voxels. But what we've pursued more recently is trying to understand how this local variability might work through whole brain or network-specific functional integration. So inspired by Carandini and Harrison at UCL, who note quite often that most of the spike invariants you can see can be attributed to synaptic inputs rather than local randomness. And they also see that spike variance is usually shared among similarly tuned neurons. So it gives you the impression that this is really more of a distributed process producing these dynamics than it is a local one. And Pincus in the 90s already noted that dynamic range of present in biological systems will suffer when you fractionate that system. So it gives the impression that we need this connection among regions to produce these dynamics that we see. So a quick cartoon of this general expectation could be if you have a node A and you have 1, 2, or 3 other nodes feeding into A here in 3 different colors. If you only have one node feeding into A in green, it should produce a relatively flat dynamic. If you have 3 nodes feeding into A, it should produce a more dynamic time series, because those regions themselves may have different time constants, they may have different neurochemical profiles. There could be a number of reasons why they don't all show up in A at the same time, but probabilistically alone, you're going to produce dynamics out of that. So the idea is that variability should rise in any region that you look when the brain becomes more integrated or functionally connected somehow. We've considered the thalamus as a key structure when linking these worlds of local dynamics and functional integration, because that region maintains projections to the entire cortex. It relays and modulates and integrates information flow throughout the entire brain. In macaque and cat visual cortices, for example, they seem to inherit temporal variability directly from those thalamic inputs. So, but the thalamus overall is rather untested as a key region when you want to link these worlds of local and sort of distributed sources. So in a first study for us, we looked at resting state data in the Rockland data set in 100 adults. It's cross-sectional data, of course. And what we noted, if you think about functional integration as measured by PCA dimensionality, if you simply line up voxels in time and do a so-called spatial PCA, you can count the number of dimensions it takes to get the majority of the variance from that data set. And if you have a lower dimensional number, accounting for all the variance there, you have more voxels talking to more voxels. So in this plot, what you see is that the lower the dimensionality is, the more functional integration there is, the higher the local variability is across the brain. And the top peak marker of that relationship is in the thalamus bilaterally. So when you see the thalamus fluctuate, really whole brain functional integration at rest is higher. If you look at the network specific level, in this case, we use the Shirer mass, so the basal ganglia network, which includes large portions of the thalamus. And you just take voxels there and you look at fluctuations in the local voxels versus the integration of all those voxels in time. The strongest within network coupling between local variance and functional integration occurs in this BG thalamic network compared to any other network. So a first pass at expressing the sort of central importance to the thalamus or the BG and thalamus in this domain. Then more recently, I'm going to talk to you about 2 studies that are still in preparation, but we have some findings to report. So the longitudinal effects, of course, are easy to summarize in the literature because there are 0. There is nothing on the variability side. There is certainly no link between local dynamics and functional integration. So we can ask questions from such data like, do adults who maintain variability over time also maintain cognition and functional integration? There's several regional targets that you might think about in general when you're doing longitudinal work in the adult population. So primary target is always a frontal cortex, for example, when looking at brain change, at least structurally so, and Naftali Raz and Uma Munenberger's work over years. With regard to bold variability, we also see frontal cortex quite regularly at cross-section expressed to be higher in younger, higher performers. And because we're interested here in the thalamus and now the BG or the striatum, of course, this frontal cortex linked to striatum, which is so well studied, cannot be disregarding the thalamic inputs there as well. So there's no striatal root to frontal cortex that doesn't traverse the thalamus. They're really a core unit, these three. So they should be considered together in mind when we look at change-change relationships. So in these data, we had 76 adults between 22 and 85 years with a 2 to 5 year follow-up at 3T, as a resting state as well. But we had 16 offline cognitive tasks. And now we do some partial least squares modeling at the latent level, just showing you the latent to latent relationship here, where maintainers of ST-BOLD over this follow-up period were also maintainers of cognition across these 16 tasks. You see the frontal cortex as a hot representation of that, both bilaterally, as well as the striatum and thalamus. And so we drill down here in the thalamus now. Remember, this is change. We use the Andreas Horn's thalamic parcellation, which is based on white matter projections from thalamus to cortex. And we show here that the prefrontal parcel from the Horn parcellation is completely dominant there. So we get the vast majority of that parcellation from this broad whole brain change effect. So we have a clear PFC bias in the thalamic representation of change in the sample. And in terms of functional integration, this change relationship I just showed you from the previous slide, the scatter is 0.39. We look at how functional integration comes in there as well. And what we see up front is that maintainers of cognition also maintain functional integration at a modest level. Maintainers of functional integration also maintain ST-BOLD quite strongly, as we always see, kind of tight coupling between these things. And that a large proportion of the delta cognition to ST-BOLD relationship can be accounted for by accompanying, by integrating functional integration into that model. So there's really a joint, nothing causal about this plot here, just a representation of shared variance sources. So it's clear that both network integration and ST-BOLD are linked to changes in cognition. Now, in a third study, because we've only looked at resting state data in the previous two, we now look at these issues, including dopamine under cognitive load. So are there task load-based change relationships here at cross-section between local variability and functional integration specific to this thalamic BG system? Can we link those associations to dopamine? And we showed in the past, as well as others, that dopamine agonism can boost ST-BOLD, but functional integration in this world has been unstudied. So beyond the BG, the thalamus also exhibits high dopamine receptor densities. So it makes sense that we look at this as a combined system again. So we have data from the COBRA study, an unusually large sample of only older adults with both 3T NBAC data and dopamine PET, showing you here the first wave of data. We have an a priori focus in this study on the thalamic BG system as a whole, where we combine Morel and BG atlases to give us the regions. And what we're interested in is this upregulation, so the modulation part of ST-BOLD here from 2 to 3 back. So what we've seen in the past and other NBAC studies as well is that most people seem to rise in BOLD variability from 1 to 2 back, at least the way we analyze it, and massive individual differences emerge between 2 and 3 back. So that's really where the action is. And people that maintain this lack of drop-off, so they don't hit this cliff and start dropping down between 2 and 3 back, usually are better in the past. So they're responding to the complexity of the task with higher dynamics. So what we see in this data set now is that the people that are able to do that, to maintain ST-BOLD and avoid this cliff, those are people with higher dopamine, higher functional integration under load, and higher speed. So that's a quick snapshot of just the BG and thalamic system, but of course we have other networks in mind that could be pursued there in the same exact way. So when we do that, looking at delta SD bold between 2 and 3 back against delta functional integration between 2 and 3 back for other networks that would be relevant for working memory, like FPN or DMN, for example. What do we see? And we find that the delta functional integration of thalamic and BG systems as a unit uniquely predicts the changes or the fluctuations in SD bold by load in every other network. So the way that bold variability is shifting in those networks under load can be attributed in a very reasonable statistical way by this thalamus and BG system. But however, the reverse is definitely not the case. So in summary, we think the first longitudinal evidence of positive change associations between SD bold, functional integration, and cognition. And this joint maintenance seems to be frontostriatothalamically dominant. The ability to upregulate variability in thalamus or basal ganglia in the face of increased task demands is a function combined of high dopamine cognition and integration. And overall, the simple take-home message is really when the thalamus or the basal ganglia fluctuates, the brain integrates both at rest and on task. So I'll be the first one to agree that we all need to study these subcortical regions a little more often to understand how the brain really works. Thank you to everybody involved in this work and my team in Berlin, and to the organizers and to OHBM as well. Thanks very much.
Hello, everyone. Thank you for joining our session on the human thalamus. I'm Kai from the University of Iowa, and for my talk, I will focus on network properties and cognitive functions of the human thalamus. So I have three main takeaways for you. First, I will present results showing that there are multiple thalamic nuclei that serve as connector hubs for functional brain networks in the human brain. So this will suggest that, yes, the thalamus is particularly important for the function of brain networks. And I will present results showing that these thalamic hubs are relevant for behavior. Specifically, if you have a lesion to one of these thalamic hubs, then that can have the potential of disrupting higher order cognition across multiple processing domains. So the first two takeaways will demonstrate the critical importance of the network position, the network topology of the human thalamus, and its importance for behavior. But we really don't know much about the functional mechanism of these thalamic hubs. And I think this is something that we know the least about. So for the final part of my talk, I'm just going to present a few ideas and some preliminary results to suggest a few potential functional mechanisms of these thalamocortical interactions, specifically for modulating functional connectivity and cortical oscillations. So the starting point of my talk is really the appreciation that when studying the human brain, we can really parcellate the human brain into several functional brain networks. So I'm sure most of you are familiar with terms like the default mode network and frontoparietal network. So you can apply different parcellation techniques and you can really extract those networks out reliably from group data or even individual data if you have a lot of data. So we also know that for this type of network organization, you do need hubs to maintain the structure and the function of functional brain networks. And applying graph theory analysis to network organization of the brain, we can conceptualize that there are two types of hub regions. There could be one type of hub region that have strong connectivity with other brain regions that are within the same network, and those will be called provincial hubs. But there are also another type of hubs that have strong inter-network connectivity. So those are hub regions that have strong connectivity with different members from different functional brain networks. And you can identify those hubs using graph theory measures such as participation coefficient. I think it's fair to say that at this point, we know quite a bit about the hub architecture and the network architecture of the cortex. We don't really know a whole lot about subcortical region. Most of the work has been largely cortically centric. So for my own interest, I was particularly interested in figuring out where does the thalamus sit in for this network organization? Is it a provincial hub or is it a connector hub? From the known anatomical thalamocortical projection, I think it's fair to say that the thalamus should definitely be one of the biggest hub region, but we want to know how does it fit into this network organization. And this work is really inspired by the framework proposed by Murray Sherman. So we know that the thalamus contains several different nuclei. We have first order nuclei, for example, the LGN, MGN, and those are nuclei that where it relay peripheral information to the cortex. But there are also nuclei such as the medial dorsal nucleus or the pulvinar, and those are nuclei that receive input from other cortical regions and then relay that input back to other cortical regions. So there is a cortico-thalamocortical motif. Given this cortico-thalamocortical connectivity motif, and then it will be interesting to see if these higher order thalamus nuclei, do they serve as connector hubs or do they serve as provincial hubs? So to figure out To test that question, the first thing we have to do is to obtain a reasonable network parcellation in both the cortex and the thalamus. And we're able to do that using public data sets. So here I'm showing you results demonstrating the typical cortical network parcellation. And in the thalamus, we're able to identify subdivisions or parcels that are strongly connected to one of these cortical networks. And then after we have that, we can then apply our graph theory analysis to see where are the cortical regions and where are the thalamic voxels that are showing high participation coefficient values. So just to remind you, participation coefficient, or PC, is a measure of connector hub property. So in the cortex, we're able to locate connector hubs in frontal and parietal association areas. This is not too surprising. In the thalamus, then it's interesting to see that these voxels are showing high PC values are primarily located in the medial thalamus, the medial dorsal thalamus, a little bit of anterior thalamus, and the posterior thalamus. You will also notice that if you look at the scale of the PC values, it seems like the thalamic voxels are showing higher, on average, higher PC values compared to most cortical RIs. And this is demonstrated in this kernel density plot. So if you look at the distribution of PC values for thalamic voxels, they are, on average, definitely higher than most of the cortical region. So here I'm presenting the same results, plotting them slightly differently. Here I'm going to compare the connector hub property of PC values for first order thalamic nuclei and higher order thalamic nuclei compared to cortical connector hubs. And you'll see that on the left, both the first order and higher order thalamic nuclei, they're showing high PC values and higher than cortical connector hubs. On the panel on the right, here I'm just plotting the PC values for a few selected higher order thalamic nuclei. The blue dashed line indicates the top 10 percentile PC values for all the cortical ROI. So that would be the definition of a cortical connector hub. And you'll notice that for almost all of these higher order thalamic nuclei, they are showing higher PC or higher connector hub property compared to cortical connector hubs. Specifically, for example, like the medial dorsal nucleus, the PO, or the pulvinar nuclei. So the takeaway is that there are really multiple thalamic nuclei showing equivalent or stronger connector hub property that are similar to cortical hubs. So now that we have demonstrated there are important thalamic hubs for the function of brain networks, are they relevant for behavior? And I think one of the stronger tests that you can do is to look at the effects of thalamic lesions. So if these thalamic hubs are really important for behavior, then there should be negative effects or negative consequences when you have a lesion just in the thalamus. So I'm going to present you some results that's going to address two broad questions. First, I'm going to look at the effect of thalamic lesions on executive function or cognitive control. And that is because we know that cortical connector hubs that are located in frontal and parietal association areas are particularly important for cognitive control and executive function processes. So if the thalamus connector hub is part of that connector hub collection, then having a lesion there should have negative impact on cognitive control. The second idea then is to test if having a lesion in this thalamic hub can impair behavior across multiple domains. And that is because the hypothetical function of connector hub is sort of cross-domain inter-network integrated processes. So having a lesion there should not just impact behavior within a specific domain, but it really should have a cross-domain impairment. And so to address those two questions, we are going to leverage the Iowa Neurological Patient Registry. So this is a patient registry that's been collected in the state of Iowa for several decades, started by the Damasio's. At this point, we have almost 1,000 patients in this registry, and they have anatomical data to describe their lesion location as well as neuropsychological assessment of their behavior. we were able to identify 16 thalamic patients from this large data set. So these are patients with, after screening, these are patients with very focal lesions just in the thalamus. On the bottom left, you'll see their anatomical images so where the lesion is in the thalamus, and we can normalize that to the MNI brain. And so on the top right, that is the overlap of these 16 patients, and you'll notice that most of them have lesions in the medial thalamus. And so we want to compare the behavior of these patients to a control group. Instead of using a healthy control group, we decided to use a comparison patient. So these comparison patients are patients also with lesions in the brain, but not in the thalamus. So we control for the lesion size. So the size of those the lesions of these comparison patients are similar to the thalamus, and they're matched in the demographics and IQ and so on and so forth. So I think this will be a better test to examine the behavior effect of thalamus lesion. So to test for executive function and cognitive control, we're going to use the trail-making test. For those of you that are not familiar with this test, there are two parts of this test, part A and part B. The part A test is really just connecting the numbers in order. You have to you give the patient a pencil, and they have to, on the paper, connect from 1 to 2 to 3 to 4, 5, 6, 7, 8. So this is thought as a control test of basic psychomotor function. The part B test is really the test that's going to involve executive function, cognitive control, or cognitive flexibility, because you can now ask the patient to connect numbers and letters in alternating order from 1 to A to 2, 2 to B, B to 3, so on and so forth. And then the dependent measure is the time that's going to take the patient to complete this task. And we have population norm to note that in the healthy population, on average, how fast someone should be able to complete this task. So after controlling for the population norm, the H effect, then we see that there's actually impairment in the part B task, but not the part A task in these thalamic patients. So that's what's the results being showing on the left. If you look at the part A result on the y-axis, if the higher, the more impairment, you'll notice that actually for part A, there's really not much difference between the thalamus patients and comparison patients. But for the part B test, the thalamus patients are statistically significantly slower. They have more impairment on the part B test, indicating that they have deficits in executive or cognitive control function. Because we have 16 patients, we're able to identify patients only with lesions that involve with lesions that involve the medial dorsal thalamus versus those patients that have lesions in the thalamus but that do not involve the medial dorsal thalamus. And we found that patients with lesions that involve the medial dorsal thalamus, they perform worse on the trail-making part B test. So they have more impairment on executive cognitive control function compared to patients with lesions that do not involve the medial dorsal thalamus. So now we show that having a lesion in the medial thalamic hub can have impairment on cognitive control function. Then now we want to know, is this impairment local to one specific processing domain, or can you actually have cross-domain impairments as predicted by the function of connector hubs? So to test that, we're going to look at a few more neuropsychological tests. So we have test results from executive function, the trail-making test that I just showed you, but we also have test results from learning and memory, language, and basic psychomotor function. For the learning and memory, we have the Ray altered verbal recall test. For the language test, we have a fluency word association test and the naming test and Boston naming test. So the prediction then is if the thalamic hub is really involved in multiple function across multiple processing domains, then we should observe impairment, not just in executive function, but perhaps in learning, memory, language, and so on and so forth. And that's indeed what we found. We found that for the visual spatial psychomotor function, as assessed by the TMT, the trail-making test part A, and the complex fluid construction test, there's actually no difference between the thalamic patients and comparison patients. But when we look at the cognitive functions, higher order cognitive functions in language, for naming and verbal fluency, learning and memory, and executive function, then across the board, the thalamic patients actually do have impairment in all of these domains. So this is really showing that if you have lesions to these medial thalamic hubs, it will be associated with behavior impairment in cognitive function across many different domains. So just to briefly summarize, the first part of the talk, I showed you the the critical network position of these thalamic regions, thalamic nuclei, and they are definitely relevant and important for behavior, because if you have a lesion in these thalamic hubs, it will have impairment across multiple processing domains. But we don't really know what is the functional mechanism of these important thalamocortical interactions, and that's something we don't really know much about, something that my lab is actively working on. So I'm just going to present to you a few preliminary ideas. One idea is that the thalamus is involved in maintaining a task-related functional connectivity structure in the cortex, given that it is a connector hub. Perhaps it's particularly good for maintaining or modulating the inter-network connectivity between functional networks when a content-demanding task is being engaged. And that's what we found. So here we analyzed a data set that we just recently published. In this data set, the subjects were asked to perform a content-demanding two-back task. And when you're doing this two-back task, that's going to activate the frontal parietal region. Then we can apply a dynamic or time-varying functional connectivity technique that's developed by our speaker, Max Shine, called MTD, that can really track how these frontal parietal regions, they're coupled together across time. Then we can actually search in the thalamus, are there regions that are showing this strong correlation between the time-varying connectivity pattern? If they are, then they're suggesting that the time-varying connectivity pattern in the cortex is being correlated or tracked by these thalamus regions. And we found indeed in the medial dorsal thalamus, there are parts of it that are showing increased correlation with this task-evoked functional connectivity pattern between frontal and parietal regions. The second idea then is to see if the thalamus can modulate cortical oscillations. So we know that when subjects are engaged in content-demanding tasks, for example, in this case, we ask the subjects to perform a switching task that can really modulate cortical oscillations. Here we found a signature of theta band oscillation in the frontal EEG electrodes. But if you have a lesion in the thalamus, so here we're showing a patient with a very specific lesion in the medial thalamus, then we test this signature in this patient, then it's actually gone. So if you have a lesion in the medial dorsal thalamus, you see this patient was not able to engage the signature of task-evoked oscillation modulation in the cortex. So that's it for the talk. So here, I'm just going to repeat the three main takeaways. The first part, we showed that the thalamus has very important network precision for cortical functional networks. They serve as connector hubs. For the second part of the talk, I have results showing that they are relevant and important for behavior, because if you have lesions to medial thalamic hubs, that can actually disrupt cognition across multiple processing domains. And finally, I present a few preliminary results just to speculate on a few potential mechanisms of what is the function of these thalamic hubs. The idea is that they could be important for modulating cortical functional connectivity, or cortical new oscillations that are important for the behavior. So finally, I would like to acknowledge our funding source and the collaborators from the University of Iowa, University of Pennsylvania, and Max Shine from the University of Sydney, and my previous postdoc mentor, Mark Despacito from UC Berkeley. And finally, if you are interested in this type of work, the lab is recruiting. We have openings for postdocs, graduate students, and full-time research assistants. You're welcome to visit our lab webpage to find out more about this. And thank you very much.
Okay, great. So thanks so much to Mac and the organizers for this great meeting and for keeping it going virtually. So I'm going to be talking about our work on thalamic control of arousal states and large-scale cortical dynamics. So the brain dynamically transitions between very distinct arousal states over the course of the day. And the most dramatic example of this is often the difference between wakefulness and sleep. So you can see in this very alert mouse on the left that this mouse is engaged, attending to its environment, whereas the mouse on the right, who's asleep, is in a very different brain state, reacting to its environment in a very different way. So ultimately, we'd like to understand how it is that the brain can transition between these very distinct states and how these in turn shape cognition and behavior. So we know that cortical dynamics are very different across arousal states. So this is now showing the EEG from a human across wakefulness and sleep. So you can see when a person is awake, their EEG has very small amplitude desynchronized dynamics, whereas a person who's in the deepest stage of non-REM sleep will have large, slow waves in the EEG. And so this overall change in the oscillatory content and the synchronous dynamics of the cortex across arousal states has been noted for decades, and is likely to play a critical role in how brain function differs across these distinct states. So while much work for decades has established the existence of a spectrum of distinct arousal states at the global level, so for example, an animal being awake or in non-REM sleep or in REM sleep, more recently there's also been a lot of investigation into the spectrum of states that can occur in cortex at local scales. So this is an example of a study in rats that was recording local field potentials in the cortex to find slow waves that appear not just in sleep, but actually even in the awake brain. And so what they found is they would see that in awake rats who are sleep deprived, there could be a patch of the cortex that would undergo a slow wave and look as if it was having a local sleep episode while other patches remain awake. And these local sleep periods in the cortex are also associated with attentional lapses and deficits in behavior, really suggesting a kind of functional consequence of these local sleep dynamics. So this line of work has made it clear that there's modulation of arousal state in the cortex, both across wakefulness and sleep, but also at these distinct spatial scales. So our lab really aims to understand what could be regulating these local and global changes in cortical dynamics and cortical state. And we focus in particular on the interactions of the cortex with the thalamus. So there's many other great talks in this session talking about these distinct structures of the thalamus. We know that they're densely interconnected with the cortex and that there's many different functional nuclei in the thalamus that may be really well positioned to modulate both state in local patches of cortex, but also in coordinating how these different cortical regions may interact with each other. So today I'm planning to briefly go over just two studies we've done in this area. The first is to ask whether thalamus might have some kind of spatial organization to it that would allow it to actually generate these states of local sleep, essentially allowing it to regulate for a given patch of cortex whether that patch is more wake-like or more sleep-like. In the second study, I'm then going to look at what happens in global changes in arousal state and say, of these many distinct nuclei of the thalamus, can we identify potentially distinct roles or distinct activity patterns across individual nuclei that may be important for regulating global changes in cortical dynamics and cortical state? The reason that we're so interested in the thalamus is because of this long body of work that has really shown a critical role for thalamus in modulating cortical dynamics. So we know that most sensory input transits through the thalamus on the way to the cortex. But in addition to this kind of forward pathway, there's also intricate interconnected loops between the thalamus and the cortex that engage in information processing. So we know that these thalamic structures are really important for synchronous cortical rhythms, particularly those that we see during sleep. And we also know, particularly from a couple of very recent interesting studies, that if you causally stimulate particular nuclei of the thalamus, you can awaken animals even from anesthesia. So a fully anesthetized animal can actually be awoken by specific focal thalamic stimulation. So this suggests that there may be really critical causal roles for the thalamus in controlling arousal state as well. And so this is what we wanted to kind of investigate further in these studies. So the first study that I'll talk about used optogenetics in mice to really causally probe different components of the circuit and try to understand the potential way in which the thalamus might contribute to local sleep. So as I mentioned, the thalamus is interconnected with the cortex. This blue shell here is the thalamic reticular nucleus, the TRN, and this is a sheath of GABAergic neurons that provides inhibition to individual thalamic nuclei. And so we thought that the TRN might be an ideal control structure for inducing local sleep because it's structured in such a way that it could potentially inhibit just one part of the thalamus with a corresponding control of state in that portion of the thalamus and that portion of the cortex. So we designed a study where we inhibited the thalamus by stimulating the TRN optogenetically. This was using transgenic mice. And we compared conditions where the laser was on, stimulating the TRN, versus where the laser was off to understand what consequences this focal TRN stimulation, so this focal thalamic inhibition, would have for cortical arousal states. So this spectrogram shows the frequency content of the local field potentials in the cortex when you stimulate the TRN. So you can see we're stimulating between seconds 0 to 30. And while we do that, there's a really dramatic shift in the frequency content of the cortical LFB. Specifically, we see that we induce these low frequency oscillations called delta waves. These are the characteristic signature of sleep. And so this demonstrated to us that, in fact, if you induce thalamic inhibition through TRN stimulation, that you very immediately and profoundly induce sleep-like patterns in the cortex. So based on this, we next wanted to ask whether indeed we could induce spatially organized control of sleep in the cortex using this stimulation pattern. So this is a cartoon of the mouse skull. This is the design that we used to test this question. The red dots show all of the different electrodes that we used to record from many different sites across the cortex. And the blue X indicates where we implanted the fiber to actually deliver the stimulation. So we're just stimulating on one side, and then we're recording across multiple cortical sites to understand where the slow waves we're inducing are appearing. So this is a bit of a busy plot, but I'll explain it briefly. So each of these dots indicates an electrode that was making a measurement, and red color indicates that we induced more slow waves. So you can see here from the red dots near the blue X in this scenario that when we stimulated just one part of the TRN, we would induce slow waves just in one local patch of the cortex. So essentially here in the ipsilateral somatosensory cortex, we would cause slow waves to appear as if that patch of cortex fell asleep. But we wouldn't see any such slow waves appearing in the contralateral somatosensory cortex, really demonstrating that this manipulation induced local sleep that was confined to just one patch of the cortex. By contrast, when we stimulated a greater portion of the TRN, now we found that we induced global slow waves that could truly propagate across the cortex as a whole. And so we would see these slow waves both in the ipsilateral and the contralateral cortex, as well as in frontal regions and other distributed regions of the cortex. So to briefly summarize this study, we found that the reticular nucleus of the thalamus contains spatially organized circuits that allow it to induce local slow waves in patches of the cortex. And so this suggests that by tuning thalamic inhibition, we're really able to induce either local or global sleep. So this points to the thalamus as a spatially organized control circuit, which allows you to have an alert cortex, patches of sleep in very spatially constrained regions of the cortex, or widespread global slow waves. And so this is really interesting to us to kind of understand this local and global control that the thalamus enables over dynamics in different patches of the cortex. One challenge, though, in this kind of mouse study is that it's very nice to have causal control over individual portions of the circuit. What we have challenges with is for measuring from more than a handful of nuclei at a time. So here, for example, we could record from the thalamus, from one nucleus of the thalamus, but not from many distinct nuclei of the thalamus to really understand each different portion of the circuit. So moving on from the study, what we wanted to ask next is how does activity across many distinct nuclei of the thalamus change as we get these dramatic large global shifts in arousal state? And so to do this, we decided to focus on the moment of awakening from sleep. So awakening can happen really surprisingly rapidly and involves a profound reorganization of cortical networks and cortical dynamics. So there's a large change in both the slow waves, which disappear, but also in just the functional properties of the cortex and its ability to interact with the world. This cartoon shows the Allen Brain Atlas of just one slice through the thalamus, showing the many really distinct nuclei that are contained within the thalamus. And these nuclei have very different functional roles. So ultimately, we wanted a way to start to look at many of these distinct nuclei simultaneously to understand how they might relate to each other as you get these major transitions in brain state. And so to do that, we moved to human imaging. So in order to image multiple distinct thalamic nuclei, we wanted to image at 7 Tesla to get really high SNR of measuring in these different thalamic structures. And we also wanted to use simultaneous EEG so that we could assess cortical oscillatory dynamics and sleep stages from the EEG signal. This study was led by Beverly Setzer, a really talented PhD student in the lab, and she's presenting this work at poster 2305 at the OHBM this year as well. So you can check out that poster for much more in-depth details and analysis of how this study was done. Briefly, the way we designed the study was to take advantage of fast fMRI to really track the timing of different thalamic signals during the process of awakening from sleep. So we were imaging subjects during sleep with fMRI using a short TR to try and track rapid evolution of temporal dynamics. We monitored arousal not just with EEG, but also through a behavioral task locked to breathing. And so this gave us a really nice tight signal to temporally lock to is the moment where people would begin performing the task after waking up from sleep. And using this, we wanted to lock all of our imaging data to that moment of awakening and ask whether there's a specific sequence of activity across thalamic nuclei that we could identify that happens at the moment of awakening. So to analyze these different thalamic nuclei, we used the Iglesias et al. Thalamic segmentation method that they detailed in this neuroimage paper, which relies on anatomical information to segment distinct nuclei of the human thalamus at the individual level. Just a handful of the nuclei that we're going to be focusing on, I'll just kind of point out here. We looked at both things like the lateral geniculate nucleus, which is a first order, lower order visual nucleus of the thalamus. We also looked at higher order nuclei, such as the pulvinar, which is a higher order visual nucleus, as well as other higher order nuclei, such as the mediodorsal nucleus, which is a higher order frontal nucleus that's been implicated in attentional and working memory processes. So what I'm plotting here now is the dynamics in just a subset of these thalamic nuclei, 4 different nuclei, locked to the moment of awakening, which is indicated by the red line. And so what you can see is there's in fact quite striking patterns of activation happening in distinct thalamic nuclei as people are waking up from sleep. So we saw really strong, for example, increases in activity in the pulvinar, the mediodorsal, and the interoventral nucleus, whereas there was much less of an effect associated with awakening in the lateral geniculate, which was our lower order nucleus. But what was also particularly interesting to see is that the timing of these was quite distinct. So firstly, you can see, for example, in the interoventral nucleus that in many of these higher order nuclei, this activation actually began to occur seconds before the person woke up from sleep, suggesting that prior to any change in behavioral state, there's actually already begun to be an increase in thalamic activation in these distinct nuclei. We also found that we could see quite consistent sequences of timing across nuclei, for example, with the pulvinar consistently leading later nuclei in this awakening process. This is further illustrated just by this cross-correlation plot that, for example, shows how these distinct nuclei's activity patterns at awakening are correlated temporally to each other. And this reiterates that there's a cluster of these higher order nuclei which are activating in concert in the moments before awakening, whereas this lower order nucleus, the LGN, showed a really distinct activation profile that, while still correlated, was kind of noticeably different from these other thalamic nuclei. So to briefly conclude this talk, we found that spatially organized circuits in the thalamic reticular nucleus can induce local sleep, allowing it to regulate arousal in local patches of the cortex. And we also found that using fast fMRI, it's possible to detect sequential timing in the activity patterns of really many different distinct thalamic nuclei that precede and predict the moment of awakening from sleep. And Beverly's poster will have lots more detail to go into on that topic. Overall, this work really argues further for the fact that there's really coordinated dynamics across the thalamus that have the ability to regulate and modulate cortical networks both at local and global scales. And I think it's going to be really interesting in future work to further dig into how it is that the complex interconnection of these many distinct functional nuclei of the thalamus and these really distributed large-scale cortical networks can act in concert to not just change these oscillatory dynamics across networks, but really then to in turn change the computational properties of the kinds of behaviors and tasks that can be executed by the cortex across distinct arousal states. I'd like to acknowledge my wonderful lab members at BU, especially Beverly Setzer, who played a big role in this work, as well as our collaborators at MGH, the Martino Center, and MIT, and funding from the NIH, the Brain Behavior Foundation, Smith Family Foundation, OneLine, the Scholar Program. And thank you all for your attention.